Hi, everyone. Welcome to our ICARE Genetics Case Conference today. Now, I wanted to start off with a case to get started. So this was a referral that I saw, a 28-year-old female who had had single-site testing in 2016 through Myriad after a mutation was identified in a family member. And there was an amended report. So this is the original report. And then there was an amended report issued in 2018. Of note, in 2021, her father had been diagnosed with prostate cancer at age 67. Now, the genetic testing result, I'm starting with this before I show the pedigree, but it was a special interpretation. And again, this gets into reduced penetrance variants. That was what the amended report showed. And here's the pedigree. So what you see here is the proband who had the mutation identified. Here is the father who subsequently was diagnosed with prostate cancer. And then this, I believe, is the original family member who had had the testing and identified to have the BRCA mutation. So of note, I contacted my colleague, who is our guest expert today, Dr. Alvaro Monteiro at the Moffitt Cancer Center, regarding guidance about this variant that Myriad calls special interpretation, but all other labs call it pathogenic. And my response from Alvaro was every bit of evidence reflected in ClinVar classification is towards pathogenicity. And this is an early truncation variant, should be as penetrant as the classic pathogenic variants. So again, pretty strong that this is a garden variety pathogenic. And I've put in here the variant as classified in ClinVar. Note that Myriad generally does not contribute to ClinVar. So again, all of them were interpreted as pathogenic. I then actually contacted Myriad, and they were fantastic with providing additional guidance about this variant. So their evidence for this variant is based on pheno, and it's expected to result in a frame shift and premature protein truncation. So on the surface, it does appear it would be a fully penetrant pathogenic variant. And they had also classified it as pathogenic before having the pheno data. And they don't know what is mechanistically going on, but there's something going on that, potentially rescues this truncating variant. So this is the reason they decide to report it as special interpretation. And they sent some curves here, which shows the distribution of why they're calling this a reduced penetrance variant. So the blue, so what you can see here, I have it all written down. The blue line is a score for this variant based on the combined personal and family histories of the individuals who are carrying the variant, and the green represents scores of thousands of matched controls. And pheno is making this a benign call compared to, they showed another variant here where, just as a comparison to show how pheno would call this one a clearly pathogenic. So basically what you see here is the red is the distribution, what I can tell here, of the pathogenics, and it's shifted to the right versus here where you see clearly it's a pathogenic variant. So again, I found this interesting because, again, sometimes we get this information, but we don't know how the call was made. So in terms of medical management for this case, the dad's prostate cancer risk sounds like it's low risk. But then looking at the pedigree, the paternal grandfather and his father both had prostate cancer, and paternal grandfather died from prostate cancer. I gave the father contact information for Mark Pomerantz at Dana-Farber to get guidance about screening for prostate cancer. Again, it's low risk prostate cancer from what the daughter was saying, but there is some uncertainty about whether BRCA1 associated prostate cancers have an aggressive phenotype. Again, implications for the family, for relatives. Maternal side, there is a strong family history of breast cancer. Cousin with breast cancer in her 30s tested negative for BRCA for the patient report. But again, during the visit, I advised that update testing may be a good idea. The family also meets testing criteria for hereditary colorectal cancer. Not sure if dad has been tested, and aunt as well had early onset colon cancer. So again, there were some follow-up items that needed to be addressed with this case. I wanted to kind of have a discussion about it alongside our guest expert today, Alvaro Monteiro, who will be presenting on BRCA variants and concepts behind what is going on overall in the field. But just to introduce Alvaro, Alvaro is a senior member in the Population Sciences Division at the Moffitt Cancer Center and a professor in the Department of Oncologic Sciences at the USF College of Medicine. He's also the scientific director of molecular genomics shared facility at Moffitt, and research in his lab focuses on applied and fundamental aspects of cancer susceptibility. Alvaro has been a close colleague of mine for years. I was at the Moffitt Cancer Center for many years, and Alvaro was one of the founding investigators for ICARE when we first started, and has consistently been my go-to to ask about these variants of uncertain significance in the BRCA genes. So with that, I wanted to go back to this case to address any questions, and also to get Alvaro's take on this variant and how we should be thinking about these things.
Thanks, Tua. I'm always happy to know that I'm the go-to person, even though most of the times you come to me, I have nothing to add. So I guess that's the kind of part of the variant analysis business. So thank you for the invitation. I think this is a very interesting variant, because it's, I am not that familiar with the pheno, and I couldn't see from the graph how many observations they had with that genotype. So, in most cases, what we're trying to do when we get to these variants of reduced penetrance, it's basically have a way in which you do a traditional case control design on a larger data set and try to estimate the penetrance from there. And I think that kind of gives me some, so we know, as I'm going to mention in some of the topics that I have today, we know there are plenty of these reduced variants, reduced penetrance variants. Most of them, however, have not been variants that are predicted to have such a dramatic effect on the protein, right? So it's an early frame shift that is truncating the protein really, really in the beginning. There's always a possibility that there may be some sort of alternative start site, that there is a transcript that is not the full transcript that is being produced, but may produce a protein that is okay from the alternative start site from then on. But it's an analysis that needs the patient's cells, for example, to figure out if this is even a possibility. Outside of that, it's really difficult. And I think Myriad recognizes that in their report or their comment to the report, that basically they don't know how mechanistically that could happen, which to me is really the big doubt for this variant, is that everything that we know so far in terms of how variants behave, that should be pathogenic. Now, and my question to in regards to the number of observations is that, it's always possible that the families or the few individuals that you're talking about has some sort of modifier or another locus in the genome that is somewhat protective and modify the penetrances. So I think that would be a possibility, but I again, without being that familiar with pheno, I would take a cautionary view of that because across the board, everybody thinks it's pathogenic.
Yeah, and that's where I think we're all recognizing it's a challenge, right? Because as clinicians, we're getting these special interpretations, yet clinically, this one is really different in my mind because all the other labs are calling it straight pathogenic and Myriad is calling it differently. It was encouraging to me to see that at least, not at least, they were transparent with why they were calling it pathogenic. So I think we all appreciate that. But as more of these come in, from a clinical standpoint, I'm not doing anything differently, right? So
I think a good opportunity for this variant would actually be some sort of functional assessment of their response to PARP inhibitors. Because I think part of what you want to derive from those information now is not only in terms of risk stratification, but also if the person has the cancer or somebody else in the family has cancer, whether that could be something that would be beneficial to have PARP inhibitor treatment. And I think we talked about this in the recent past too, about the challenge of all these reduced penetrance variants, because there is quite a bit of correspondence between risk and response to PARP inhibitors, and carriers of these reduced penetrances may have this kind of duality in which, as far as risk is concerned, they are on the lower bound of the higher risk, if you will. But if they get the cancer, they may have less therapeutic options than a carrier of a traditional truncating mutation, for example, right?
So are you saying, again, I remember we talked about this, and it's fascinating, right? Because the studies could actually be done because many breast cancer survivors or many breast cancer patients with a BRCA mutation are getting PARP inhibitors at this point, just with the widening recommendations like the FDA approvals. So
I do think that perhaps the ovarian cancer setting might be more informative, because the way that now PARP inhibitors are coming to be frontline treatments more than basically
compared to breast,
only the metastatic of people that already failed many other treatments. And one of the complications that we have seen is, again, it's not a complete correlation, but there's quite a bit of correlation between platinum resistance and PARP inhibitor resistance, so that sometimes what we see in a patient that is, we think it's just innate resistance to PARP inhibitor, in truth is actually an acquired resistance that the cell acquired not because he was treated with PARP inhibitors, but because he was treated with cisplatin, for example. And that led to the same mechanisms that are selected in the tumor that would now already make the PARP inhibitor a not so good option as you would have in the front line. So that's why I think that is something that is interesting. I think this could be done on a cell on an in vitro basis, at least to get some idea of what's going on. But it's always challenging because you have all of the in vitro systems that we have right now. They may have issues at actually looking at PARP inhibitor sensitivity in a way that we can, right? So let me rephrase that. I think we've now grown accustomed to say that a defined pathogenic variant predicts response in PARP inhibitor to a certain extent, but the reverse, meaning can we use the information of PARP resistance in the clinic as a way to verify the classification of a variant? To me, it's one of the most fascinating questions that we can address right now. And we've been trying to do this with real-world databases, such as SciAPSI and Flatiron, for example. But I can say already that the numbers are not there yet. There hasn't been enough carriers treated with PARP inhibitors for us to ask these questions. But basically, for these databases that you have patient trajectories after treatment, you could basically devise a way in which now knowing which variant is being carried and how that patient behaved in terms of treatment, that could also be a line of evidence to classify a variant that may be a VUS.
Yeah, no, that is fascinating because I don't think we're collecting it in that way at this point in time. So really important. I don't want to shortchange your presentation. We have a couple more cases that are prepared. So I'm going to let Lindsay hand over to you.
Okay, so thank you very much for the invitation. I think I always like to start with this, one of my favorite pop artists, Jane Rosenquist, who was a contemporary of Andy Warhol and has this beautiful painting that, in my view, always represented a little bit the knowledge of variants in BRCA1 and how they relate to risk. If you think about how we were at the very beginning, at the top of this painting, in which, by the time we started cloning BRCA1 and BRCA2, we had all these jumbled information about how to stratify patients for risk. And then eventually by cloning and the genetic testing becoming available and we knowing a lot more things about it, then the picture becomes clear that now you can figure out the numbers on the dial, if you will. But then, the other thing is what's happening right now is an explosion in variants, variants that also may have a role as somatic variants, not only as germline. The realization of the spectrum of effects that takes into consideration not only the highly penetrant alleles that we cloned in the families that were selected for linkage analysis, but also variants that have reduced penetrance like the one that we possibly that we discussed a while ago, but also with the deluge of genome-wide association studies and the development of polygenic risk scores. It looks like the waters are becoming a little bit muddier again, but my feeling is that this is very temporary, and I think we're going to very soon be able to have a much clearer picture. So let's see how I am trying. I do not seem to be able to move the slides.
If you click on your screen, Dr. Monteiro,
Yes.
You should be able to take control.
Okay. So just competing interest is disclosure. I have no stock or stock options or any relationship whatsoever with any healthcare, precision medicine, or diagnostic companies. And I think that's an important thing to disclosure. And there's a few others that you can see there as well. So what I like to do today very quickly is basically talk a little bit about the notion of the spectrum of allelic effects and how we have to think about a world in which we're not only deciding between a pathogenic variant that will trigger recommendations for prophylactic surgery, for example, and not pathogenic, but rather, the spectrum of really different effects on risk and how we can come up with ways to customize what kind of clinical recommendations we may be able to give to women individuals that have a different estimate of risk in terms of the balance between the benefits and the risks and comorbidities involved in some of these choices. I'm going to talk a little bit about the classification schemes that are now used to classify variants. My focus has always been on functional assays, and I'm going to try to put them in context and touch a little bit on the latest projects that we have on functional data integration and how we can now arrive at a lot more information for variants than before there was none. I'd like to touch a little bit on the role and the importance of data visualization and tools to do data exploration. We're going to talk a little bit again about intermediate and low penetrance variants and how to try to deal with them, as well as some new trends. Please raise your hand or unmute and say something if you have questions. I'll be happy to answer them. So I think, I always like to start with this graph from Terry Manoliu's paper that basically shows what we think is the model of allelic structures for most of the disease genes that, sometimes we call it the common variant, common disease model. But the idea is basically that there is an inverse relationship between effect size and frequency. Variants that have a lot of impact on risk will normally be kept a very low frequency in the population. Variants that have very minor effects on risk will be able to fluctuate and reach much higher frequencies in the population. So the first group of variants that we really identified as a field, and then those were the variants that are really the target of genetic testing, are what we call Mendelian. Just in a way, basically, the idea is that it's very high penetrance, so that most of the individuals that carry the genotype will develop the phenotype, and that's the basis of you being able to use affected families to do linkage analysis. And the result of that was really the identification of several alleles that change the protein coding for BRCA1 and BRCA2. And a lot of the things that we're going to talk about today are going to be relevant to BRCA1, BRCA2, and BRCA3, or as somebody calls them, PALB2. So basically, the idea is that these are variants that are very rare, right? And they have very high effects on risk, 10-fold, 7 to 10-fold increase in risk of breast cancer, about 30-fold or a little less increase in risk of ovarian cancer, very, very strong effects on risk. So these variants come in a number of different flavors, and what you see here is basically kind of the BRCA1 locus idealized, if you will, that you have all sorts of these different variants, regulatory site variants, missense variants, splice site variants, splice site that lead to alternative exons, nonsense in frame sheet that lead to premature termination, and not even depicted here are large rearrangements, for example. And what you can see is basically that it will be a number of different effects on the protein. Some of them, it may be translated in a smaller amount of the protein instead of a larger amount of the protein. Some of them may be a different protein will be produced that has a different format or things of that sort. So there's a number of things that we can look at. And the expectation is always that variants that are related to increased risk are variants that are going to knock out or severely knock down the protein function, either because no transcript is produced, or the transcript is defective, or the translation is defective, or the protein produced is defective all around. And normally there is several ways that we can define whether a particular variant is going to be pathogenic or not. And some of them we can simply infer from sequencing, right? So that's the case for most of the variants that are going to lead to premature termination. Interesting is that the case that we just discussed seems to be one outlier of this class of variants that in most cases, those are variants that we call sometimes legacy variants, because we know if it's a premature truncation even of a few amino acids at the end, it will lead to a non-functional protein. We can use allele frequency in the population, going back to the graph that I just showed you, suggesting that if something has a frequency that is very high, and by very high, the threshold is normally around 1 to 2%, depending on each outbred population. But normally if a variant is found that frequencies are higher than those thresholds, they are unlikely to be pathogenic. But remember, there's always the confounding of populations that may have some sort of founder effect, in which you may find some pathogenic variants at frequencies that are unexpected, right? And then there's a number of ways that we can do this by looking at the molecular biology of all of these transcripts and proteins, and try to figure out whether the protein is still functional or there is a significant impact on function that would correlate with risk. So normally what we have here is basically that we use several sources to classify these variants. I just mentioned to you the ones that are inferred by the genetic code. We can also use segregation analysis and looking at different pedigrees. Also we can use association studies, case controls, for example. And we normally, in the case of BRCA1 and BRCA2, we also use co-occurrence, because we know that embryos that lack both copies of BRCA1 or BRCA2 are normally not viable. And if they have some residual, in the case of BRCA2, they may cause Fanconi anemia as well. But it's normally, by definition, if you have a variant of uncertain significance that co-occurs with a known pathogenic, this variant of uncertain significance is very likely to be benign. So as you see here, a lot of these sources of evidence were really incorporated in a statistical model developed by David Goldgar and Doug Easton, in which all of these informations would actually coalesce in this model. The problem with this model is not the model itself, but the fact that almost every source of information depends on variants and observations that are normally very rare. right? So if you want to do informant segregation analysis, given the vagaries of obtaining families and testing all individuals and individuals that are informative, it's very hard to have several families from the same individuals. Association studies is the same thing. We're going to talk about some of them, but in general, because the frequency sometimes is less than 1 in 10,000, you really need very large data sets to be able to do that. So the multifactorial model is unbelievably important to coalesce all the information from the several sources, but still leaves many, many variants without classification. And the way classification is today, in most of the academic centers and most of the companies, they basically follow a very similar framework in which you have several tiers of classification, as shown here by the Enigma framework. And basically, what you have is a scale of 1 to 5, in which we start with 1 at benign, 2 likely benign, 3 variants of uncertain significance, 4 likely pathogenic, and 5 pathogenic. I think the important thing to stress here, and probably the biggest misunderstanding of this framework, is to think that the numbers have any relationship to the severity of the disease caused by the variant in question. A variant that is class 4 is not less severe than a variant that is class 5. The only difference is that the data, the certainty that we know that this is pathogenic is less with a 4 than with a 5. So the only thing that changes is not the severity, but is the certainty that we have in the information available. So that's kind of very important. In general, most of the companies will have some sort of variation along those same themes. And the idea is basically that these variants would be classified, in the case of Enigma, by the multifactorial model, and they would be deposited. For many years, they were deposited at the BIC, the Breast Cancer Information Core, at the NHGRI that was spearheaded by Larry Brody. We eventually transfer all the information from BIC to ClinVar, and today, all of this data is in a very nice way, all coalesced into BRCA Exchange that I really do recommend that you all have access to. There's also, I don't think it's advertised, but there is a phone app for BRCA Exchange, and you can also create alerts if you are tracking a certain variant and get an email when something has changed. So, if we think about this today and we you think about the television newscast in the late 90s or the mid 90s when BRCA was cloned, the impression that we had was basically that you had a good gene and a bad gene, right? So it's like 2 different forms, a pathogenic and a benign. But what we know is basically that there are lots and lots and lots and lots of forms of BRCA1 and BRCA2. So today, documented, meaning that found in the population, we have already listed 68,000 forms of BRCA1 and BRCA2. There's about 34,000 of each one. And there are all types of different variants. And of course, many of them are classified benign because they are high frequency and they're not really an issue. Don't get scared by the number of variants. We know pretty much a good size of these variants, what they are in terms of risk stratification. So one of the problems with the multifactorial was also that we didn't have ways to incorporate functional data, for example. And in many cases, there were other types of data that were also not in the multifactorial data model. And now this was kind of resolved in some way by the publication of the ACMG AMP rules, which are shown here, that created a number of categories of lines of evidence that go from supporting, moderate, strong, and very strong. And they're really calibrated and they generate codes, as you see here. So that allowed for the introduction of many more sources of variation in a model that can be applied by anyone if you understand the codes. So we've been working quite a bit on functional analysis for many, many years. And the idea here is basically to create a test in vitro that can verify whether the protein is or is not working well. And these tests are independent on whether they focus on a specific known biochemical function of the protein, but it may be a surrogate of that function. And if the specificity and sensitivity are high enough, then we consider that a good thing and a test that can be used. And one of the things is really try to do data integration, in which you're now looking at different data sets from different independent labs that have tested the same variant and really be strong in numbers and be able to integrate all of that data. So we've been kind of setting up the stage to actually not only for us to be able to integrate the data, but for anyone that wishes to do so, to do that. And the first thing that we did was the creation of the BRCA1 circus, which was at the NHGRI BIC. And you see here just a little bit of an inset of that. And basically, if you think about this, this is every single variant is depicted there, and every single dot means a source of data that you can click on the data and go to the original source. And at that same time, be able to look side by side, not only individual variants, but also to have an idea of how individual regions of the protein may behave and be more or less tolerant to mutations. This was up until, I think, last year, where the BIC was deactivated. You can still have access to that. We now transfer that to the FYI HBOC, which is also a repository of all data, functional data for BRCA1, CHEK2 and PALB2, and BRCA2 is also coming up. So that's the idea, is now that you have to put all of these things together to be able to make sense out of it. So the first thing that we did after doing all that was to basically take all that data, and we were able to identify several dozens of different tests for BRCA1 and BRCA2, and find out that there was information for over 2,500 variants in BRCA1 that were classified as VUS. And we were able to do, using the integration of the data, to provide evidence for classification for about 2,355 variants of uncertain significance according to their pathogenicity, using exclusively the functional data. So there's a huge debate about whether having just one source of information is enough for us to make a call, but that's something that we're slowly building into the model as well. One of the issues that we've had is basically when we do any type of functional assay, where do we draw the line? And basically, the big thing is that you can do it in any form. You can do a linear regression, you can do standard deviations from the wild type, you can set an arbitrary threshold. So we developed VARCOL instead, which is a brainchild of Ed Iverson. And most of the things that I'm telling you today, it's been kind of a very strong collaboration that I had with Fergus Couch, Ed Iverson, and David Goldgar as well. And the idea basically here is that the model is able to use the data and do two things. The first one is to define the probability of pathogenicity of each variant, depending on each activity, but also because the model changes at every data you add, you actually refine the classification of the variants you tested before. So it's sort of a living model that we think will have a lot of use in this. Coming out tomorrow is the extension of our call that now can put together not only the functional data, but also family data and protein predictor data. And I just want to spend 1 second on this graph for you to have an idea of the difference of the functional data and classification. So if you look at the graph on the left side, this is the null model. Basically, all these variants are unclassified and they remain in this white region of the graph. And as you add more information, you can see that the lines, which represent individual variants, start falling into their categories. The green one is the benign, the red one is the pathogenic, and the green lines are the known benigns, and the red lines are the non-pathogenics. And what you see basically here is that the addition of functional data makes, first of all, if you have a model that is functional data only, you already obtain a very nice discrimination between pathogenic and benigns. But, you know, as you add family data, you get also an improve in the classification. The issue again is that the family data is very, very nice, but there's not a lot of family data for many of the variants that we wish. So having functional data only is already a powerful way to classify these variants. So, as you know, we look for what we call white eye mutants. So this is a reference to Thomas Hunt Morgan, the father of genetics, that found a white eye mutant, and that's how they started mapping genes in a linear pattern as we do in linkage analysis. And so we're always trying to look for outliers, and eventually we found a couple of outliers. And these outliers were outliers that either had been previously classified as benign because of co-occurrence, but the functional data suggested that they were pathogenic, for example, right? And they were considered benign because they co-occurred with another pathogenic variant. To make a very long story short, what happens is the example of one hand doesn't know what the other is doing. Normally, the co-occurrence data shouldn't have been used because that family actually hid some sort of syndrome that was similar to Fanconi anemia type of, in which the patient had learning disabilities and many other hallmarks that family was not normal for the purposes of calculating co-occurrence. And a beautiful detective work led by Roger Greenberg to actually find out that some of these variants were indeed pathogenic, although seemed with a reduced penetrance or a partial function. So that kind of led us to the realization that where there's one, there's probably more. So we need to deal with these variants as well. So again, we're now moving to other groups in that spectrum. One of them is this low frequency variants with intermediate effects. Sometimes the effect is so low that you don't see the patterns in families, so you resort to a more traditional case control design, and eventually identify that they do have intermediate penetrances, and this is shown here by two analyses that we performed led by David Goldgar and Fergus Couch, looking really at using the collaborative oncological gene environment study data from the GWAS to actually estimate the odds ratio. And as you see there, you see lots of variants that have odds ratio that are moderate, to say best. So there's definitely a lot more of these variants out there than we'd like, because they are problematic for us to tie them to clinical management options. So the new trends is basically, how can we generalize this to other predisposition genes? PALB2 is what comes to mind initially. How do we do the integration and the classification of variants using VAR-CAL and ACMG rules? How do we deal with intermediate and low penetrance variants? Remember, at that one of the first slides, I said that one of the biggest misconceptions is to think that the numbers correspond to the severity of the disease. And in this case, basically, we need to modify the model so that the severity or the penetrance is now built into the model, so that individuals can have better risk stratification estimates. And that is very similar to what we're going to do with common variants generated by genome-wide association studies and polygenic risk scores. So just to finalize, here is one of our efforts in PALB2. We had a very large consortium of PALB2 that was part of the PALB2 interest group led by Mark Tishkovitz, that we kind of had a subgroup of people interested in function and looking at missense variants in PALB2, and also creating a resource where we have these variants out there for people to take a look at and also do their own analysis. And again, how do we deal with these common variants? Variants that instead of increasing by tenfold your risk of cancer now increase by 10% to 50%. And they're very important in the case of polygenic risk scores, because we know that even if polygenic risk score may not change much the estimate of the risk for many individuals, there are certainly individuals at the top percentiles of risk that, because they accumulate many alleles that have small effects, their combined effect may be as large as a traditional truncating mutation in BRCA1 or BRCA2. And I am going to stop here and just hear some of the people that have been participating in this effort that's taken many years, with a lot of thanks goes to Marcelo Carvalho in Brazil, Ed Iverson, Fergus Couch, and David Goldgar, as I said, and also Marcy Richardson from Emory Genetics, who is the repository of knowledge of all the ACMG rules. So if you have questions about that, ask Marcy. And I will stop here. And I just say that I think in the presentation, my email and my Twitter is there. So feel free. I think Tua knows that one of the things that I've committed a long time ago was to respond to emails from genetic counselors and providers on a 24 to 48 hour basis. And I've done over 500 of them, and I'm pretty happy. Actually, they have triggered many studies. We now have a study together with ICARE that we're looking at incorrect extended tails of BRCA1. So there's a special class, but feel free to email me if you have questions.
Thank you as always. I will continue to reach out to you as I always have. Thank you everyone for your attendance, and we will see you next month. And thank you again, Alvaro. Bye.